Hi, Miss Brink. I'm outside and it's really cold and it's windy. And if you hear people screaming, it's because my cousins are out here with my sister. Anyway, I've watched the Mr. Anderson's video. I did the lab online, the virtual lab. I took the self quiz and I got all the questions right except for one of them. But I went back and I read it and I realized what I did wrong. I corrected it and I got it right. And this is what I've typed out what I'm gonna say because otherwise I'll be sitting here making you feel awkward and stuff. So I'm just gonna yeah. Anyway, okay. This new lab is to help us learn how to measure the rate of respiration in a living organism. To start off, we have to fill up the bottom of the respirator, which is the glass tube part, with potassium hydroxide. Then place an absorbent cotton ball to help absorb it, and then put a non-absorbent cotton ball on top so that the organism, plant, the pea, or the grasshopper, or cricket, whichever one you choose, doesn't get harmed. After the cotton is in place, we will put the organism in, followed by the stopper. We'll be doing 3 respirators. One will have if we do peas, one will have the germinating peas, one will have non-germinating peas and beads to make sure that the volumes are the same, and one will have plain beads as our control. Once the stopper's on, you'll put the weight on top of the pipette so that it stays down in the water. After it's all done and put together, we will place it on an angle in the tub so that it can equilibrate. There's going to be a 10°C bucket and then a 25°C bucket. I believe it's 25. It might be 20. The end of the pipette will be out of the water so that it doesn't actually start the experiment. It's just going to equilibrate so that the temperatures are the same, and so we know what to account for with the temperature change. That way our results aren't skewed. After the equilibration period is over, one of the team members will gently place all of the respirators into the water, and once the pipettes hit the water pressure is going to help form an air bubble from all of the oxygen. So there's going to be an air bubble, and that's what we're going to be using to see the rate of respiration. Get out! We measure the placement of the air bubble in intervals of 5 for 15 minutes, and once the data is collected, a graph will be made by either me or one of the other members. Once we get the lines, the slope of the line, each line for the 3 respirators, those slopes represent the rate of respiration. And that is it. So, yeah, bye!